Yes, are you able to see the slides?
Yes, ma'am.
Yes, ma'am.
Yeah, so last class we were seeing about this diffusion, osmosis, okay, facilitated diffusion, and then active transport. Okay, so active transport, I told you, it's the transport, okay, across the cell membrane through carrier proteins and against concentration gradient. Okay, and it requires energy. Sodium potassium pump is what is an example for what this active transport. Okay, then we saw the summary of all the four means two kinds of transport and their different types. Okay, so next I think I started with nucleus. So what all we have seen in nucleus till now? Is what all the information I have told you regarding nucleus till now?
Ma'am, that it is a double membrane structure
Yeah.
and it contains all the genetic material, and about the DNA and RNA, and we were discussing about the helical structures and the DNA part.
Yes. Anyone else?
Nuclear pores
Mm. Yeah.
and the structure of nucleus,
Mm.
RNA, DNA.
Okay, so what we have seen is that, see, like we have seen how actually nucleus is. So it's just like you have told, it's a double membrane one. Okay, so the inner membrane, it covers the nuclear part. Okay, so it keeps it isolated from what? The cytoplasm, and the outer membrane, it will be in contact with what? Other organelles like endoplasmic reticulum, etc. Okay, so this is regarding your nuclear envelope, and I have told you the composition. Okay, so it's a phospholipid membrane only. Okay, so here also, just like your cytoplasm, the nuclear membrane is also what? It's made up of two phospholipid bilayer. See, but the proteins, okay, whatever it was there in the cell membrane, and the protein here in the nucleus, it may vary. Okay, but otherwise, in general, it is composed of what? Phospholipids and what? Protein. Okay, but the protein, whatever it is going to be there in the nuclear membrane and the cell membrane, it will vary because the function varies, right? So that it varies. Otherwise, the structure, it is going to be what? Same. Okay, so next you have nuclear pores. So the transport between the cytoplasm and the nucleus, it will happen only through what? Nuclear pores. Okay, so sometimes RNA, it has to travel at ribosomes, it has to see RNA, ribosomes and all, it will be synthesized within the nucleus. Okay, so whatever it is synthesized, that it has to be taken to what? The cytoplasm. Okay, similarly from cytoplasm, some materials, it can move inside what? The nucleus. So the nuclear pores are the openings on the surface of the nucleus, which actually it controls, okay, this what is that? Controls or regulate the exchange of materials. Then you have something called as what? Chromatin. Okay, so I have told you chromatin threads are what? Long strands of DNA and proteins. Okay, see, I don't know, do you see, have you studied, at least I don't know, in 10th standard, the structure of DNA, have you studied?
Yes, ma'am. We
Huh?
had studied in 10th standard.
Structure of DNA.
Ma'am, I
Yes.
only remember prokaryotes.
Yes.
Prokaryotes and eukaryotes, it was right from 6th to 7th, I think, no?
That's
Yeah.
all, ma'am.
It's okay. It's okay. See, listen. Today again. See, for your syllabus, actually, exactly what actually is DNA composed of, and what is this chromatin composed of, are chromosomes in detail. Nothing is needed. Okay, but you should have a knowledge. Okay, you should know at least what actually is a DNA, what is actually a gene, what is this chromatin, what is this chromosome. All these things, it's good for you to know and keep. Okay, so for that purpose, I will play you a video which shows, okay, exactly the structure of a DNA. Okay, and one more video I will show you which shows how actually proteins are synthesized from what? DNA. See, normally they tell nucleus, it contains all the genetic information, they will tell. Okay, but how actually these genetic informations are useful? You know, see this heredity, what they will tell, it will be carried from generation to generation. Okay, so everything, okay, everything in the form of codes are in the form of nucleotide bases. It is stored in what? DNA. Okay, so this DNA is the one which is going to control the synthesis of protein, or it is going, see, protein is what, this combination of protein only is what is going to decide, okay, a cell. The cells combine to form what? Tissues. Tissues combine to form what? Organs. And finally, organ gives you what? Full entire structure, right? So if you see, DNA, it is going to be the basic, and the information present in the DNA, it will decide which kind of a protein to be synthesized. And what actually, see, protein is just nothing but what? It is a sequence of amino acid. Okay, I've already told you it's a polypeptide chain. Okay, polypeptide chain means what? Amino acid linked to each other. Okay, finally, these amino acids, it will undergo some structural, what is that, changes, and finally it will become a protein. Okay, but otherwise, in simple words, it's a sequence of amino acid. So suppose some protein, okay, or some enzyme, for example, insulin, something if it has to be produced, okay, it needs, okay, which all amino acids and in what sequence it should join, okay, that information is what is provided by, it will be provided by your what? DNA. Okay, so that much important is going to be your DNA. So if you see nucleus, it controls all the metabolic activities, okay, growth, all metabolic activities, it is controlled by what? DNA. Okay, so majority, okay, of the DNA is present only in the nucleus. Okay, but do you know any other organelle present within the cell which will have DNA? Any other organelle which has DNA inside the cell? Anyone? Any idea? Any idea? Anything you know?
Mitochondria.
Yeah, so it's going to be mitochondria. Okay, see, mitochondria, it's the other organelle which has some DNA, but not like nucleus. But that DNA, it will be used by mitochondria to produce its own protein for its own, because mitochondria, it is going to produce what? ATP. Okay, see, that is what is going to give the energy. Okay, so some protein, whatever it is needed for its own function, okay, that it will be produced by the DNA present inside what? Mitochondria. Okay, so nucleus is composed of what? Complete DNA. Okay, majority it is present in what? Nucleus, but mitochondria is the other organelle which will be having some DNA. Okay, needed for its own purpose, for its own thing. Okay, so chromatin, and next I told you nucleolus. Okay, so nucleolus, this is the one which is going to make rRNA, I told. Okay, a ribonucleic acid. Okay, a kind of rRNA, ribosomal RNA, and even ribosomes. Okay, these, so nucleolus, it's involved in the synthesis of what? RNA and ribosomes. So these are some of the important things in nucleus, okay, which is very much needed for your thing at least. So I think I have shown you all these things. So these are the nuclear pores. See, are you able to see here? These are the nuclear pores. Okay, so this is what is going to allow the transfer of RNA, all those things present. So they have marked here, see, this is what is going to be the nuclear pore. See the actual diagram, whatever they have shown, the black and white thing here, whatever they have shown, no, this is the original thing. See here you can see this is what actually is the pore. Okay, and I have told you some ribosomes are also, see, ribosomes will be synthesized inside the nucleus, and some ribosomes it will be there on the surface of the nucleus also. That is what they have shown here. Okay, ribosomes. So here they have seen shown. See, this is actually the outer membrane and actually two membranes, right? Inner membrane. See here, there is they have introduced one more terminology called as nuclear lamina. See, this is what which provides support to the nucleus. Understood? See, in the case of cell, I have told you cytoskeletons, okay, microtubule, microfilament, all those things, it will provide support to the cell, cytoplasm as a whole. Okay, but in the case of nucleus, it is going to be what? Nuclear lamina. Okay, nuclear lamina is the one which is going to provide support. Yeah, so now, can you close the door? Yeah, so now this shows actually what actually it's a DNA. Okay, and see actually chromosomes you might have seen. No, here this is actually not a chromosome; it's a chromatid. You will remember it like this, right? This is what is called as a chromosome. See now, it will be like so many threads of DNA coiled together, right? See, it will be like this, and I have told you this chromosomes it gets formed only when the cell is about to divide. Okay, otherwise you can see this in terms of what? Chromatin fibers only. Okay, here they have shown the difference. You see this is what actually is the chromatin fiber. Okay, normally it will be like this only, but when actually this chromatin fiber, okay, during that is when the cell it's about to divide, okay, it will just coil itself like this, and so many coils are you able to see this is a chromatid. Okay, this together two sister chromatids together is what is called as a chromosome, but just for diagram purpose they have shown it like this. Okay, so chromosome is what? It's a tightly, okay, condensed packed chromatin fibers. So now if you see what are these chromatin fibers, these are the chromatin fibers. And what is this chromatin fiber made up of? It's made up of proteins. Okay, particularly this protein, it will be called as what? Histones and a DNA strand. Okay, so this is what is the DNA strand. So what is chromatin fiber? Chromatin fiber is composed of what? A DNA molecule and proteins. Okay, and this when it is packed in a very condensed form, okay, during when the cell divides, it will be called as what? Chromosomes. Okay, now what is actually a gene in this? Anyone? I told you this is a DNA molecule. So, and DNA molecule combined with chromosomes, it's sorry, protein is what is called as a chromatin fiber. Normally, if it is just scattered like this, means it's a chromatin fiber. Okay, but if it is tightly packed, condensed form is what is called as a chromosome. Okay, now in this, which is actually called as a gene,
Proteins, ma'am.
proteins, no, which is actually called as a gene in this figure at least, if I have to ask you. See, this is purely
Gene isn't gene like the combined like DNA makes up gene, right?
yeah, see, but just nothing but gene. But see, for example, suppose the functional unit of a DNA molecule, or else this complete strand, okay, or else see which contains, okay, the information that it can, using that information, if you're able to convert that into a protein means, then that particular functional unit is what is called as a gene. I can call this also as what? A gene. Okay, similarly, suppose if you from here till full entirely, okay, this also can be called as a gene. Suppose if this can code into what the information stored here, if it can get converted, okay, into a protein means, then that also it is called as a gene. Okay, so the functional unit of a DNA molecule is what is called as a gene. Understood? So this is what is called as a gene. Okay, gene, it's just nothing but what? A DNA molecule only, a fragment, a segment of a DNA molecule is what is called as a gene. Understood? See now, what actually is this DNA molecule made up of? That is also there, right? See, are you actually this structure, if you remember, it was proposed by what? Watson and Crick. Okay, see, those are the people who actually brought revolution in the field of biology itself by actually giving this structure. So you have a double helical structure. Okay, you have two strands, right? One strand running in this direction and another strand running in what? Opposite direction. This is what is called as a double helical structure. See, this particular blue thing, no, this is made up of sugar and phosphate. Understood? This is sugar phosphate backbone, normally they will tell. Okay, this is what? Sugar phosphate backbone. And this stick like structures, what you are able to see, no, these are just nothing but nitrogenous bases. This is what is your, you have, we have already seen, no, adenine, thiamine, okay, quinine, then cytosine, ATGC. Okay, so these are the nitrogenous bases. Okay, see, these bases will be connected by what? Hydrogen bands. Okay, hydrogen bond. Okay, see, neatly they will, I will show you a video where they would have explained very neatly everything. Okay, so now first, okay, you have DNA. Okay, what is DNA? It's a double helical structure where you have a sugar phosphate backbone, okay, which is connected with nitrogenous bases. Okay, so these are the bases. There are only four types. ATGC, this only often it will get repeat itself, and the, okay, I will explain. Okay, this is what will get repeated itself. Now this DNA, when it combines with proteins, okay, then that is called as what? Chromatin fiber. These chromatin fibers, okay, finally in the condensed form is what is called as the chromosome. Understood? So now I will play you a video. See, now let me tell you, the video is just purely for your understanding purpose. Okay, from exam point of view, only this much is enough. You just know what is the DNA, what is a chromatin fiber, what is a chromosome. Other than that, whatever it is explained in the video, it's only for your knowledge. Okay, it's not for exam purpose, so don't get worried too much by the formulas, whatever they are showing, all those things. Okay, it's just for instructional purpose. Okay, let me make it clear. Everyone, is it clear? Okay, the whatever video I'm going to show you now, it's because it has too many formulas, that bond, this bond, like that, so many things it will come. So don't get worried by all those things. It's not for exam purpose, just only for your understanding. Okay. Yes, are you able to see the screen?
Yes, ma'am.
Yes, ma'am.
This is
Is it audible?
Yes, ma'am.
Yes, ma'am.
Okay.
DNA, or deoxyribonucleic acid. By the end of this video, you will be able to identify the key structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details, but all views represent this same structure. Inside, we find double-stranded DNA, in which two strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also called B-form DNA. Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has three components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine.
See, are you able to understand? See, DNA, it's just nothing but a polymer of nucleotide. Okay, so what does this nucleotide means? It will have a sugar group, a phosphate group, and a base attached to that. Okay, and that base, it can be anything: adenine, quinine, thiamine, and cytosine. Okay, so this is what is called as a nucleotide. Okay, so many nucleotides combined together is what is called as a DNA.
Guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT, or TA, and GC, or CG, base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the base is attached to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions form when the aromatic rings of the bases stack next to each other and share electron probabilities. The regularity of the helical structure forms 2 repeating and alternating spaces called the major and minor grooves. These grooves act as base pair recognition and binding sites for proteins. The major groove contains base pair specific information, while the minor groove is largely base pair nonspecific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact with in the grooves. In this way, the DNA can be acted upon in either a sequence-specific or non-sequence-specific manner, allowing proteins to position themselves correctly in the genome to carry out their designated tasks. This is the DNA double helix, and you've now learned the structural features that influence its function. We hope you've enjoyed exploring this.
So, was that clear how actually the structure is? Okay, so I have told you, right? It is the backbone. It's made up of what? Sugar phosphate. Okay, and attached into what? Four nitrogenous bases. So sugar, phosphate plus one base is what is called as a nucleotide. Okay, so you have DNA. Okay, it's just a strand of what, or a polymer of what, nucleotides. So two strand like that, it will get intertwined. Okay, to form what? DNA. And one more important thing, nitrogenous bases. Okay, they have told, right, C and G only can pair, A and T only can pair, because the other two pairs are not possible, because they have told no, the geometry will get disturbed. Okay, so the way actually these nitrogenous bases are arranged, so this is what is actually very very important. This is what actually it will code for what, the sequence of amino acids. Okay, see normally this 3 3. Okay, normally they have told in a turn there are 10. Okay, 10 bases, right? So 3 3 together normally it will get read. Okay, and that actually it will get converted into what, an amino acid. So that is what is actually called as a codon. Codons means what? Three, just consecutive. Okay, next three. Thing. Yeah, so now is it clear? DNA molecule. Okay, so these things are just nothing but what? Nitrogenous bases and sugar phosphate thing. This when combined together with protein, okay, it's what is called as a chromatin fiber, and that in the condensed form is what is called as a chromosome. Okay, now coming to the functions of the nucleus. Okay, so we know, right, what will be the main function? The main function of the nucleus it is to control gene expression and facilitate the replication of DNA during cell cycle. See, just because DNA it is there, okay, it controls almost every activity of the cell. Okay, so it controls all the metabolic functions of the cell, like growth. Okay, everything, cell division, everything, it will be controlled only by what? The nucleus. Okay, so the nucleus controls the metabolic functions of the cell by producing mRNA, which encodes for enzymes, example insulin. Okay, see this, how actually a protein it is going to get produced is what is very very important. Okay, so that also not in depth, let me just tell you how actually it will get produced, because right in next we will be studying about ribosomes. Even in ribosomes also the same topic it will get repeated. Okay, now just let me tell you in a very simple form how actually the proteins will be produced. Later for that also I will show you a nice video whereby seeing you will be able to understand. Okay. See now DNA, it's a double-stranded sugar phosphate backbone with a nitrogenous basis. Okay. And why it is called as deoxyribonucleic acid, that also we have seen. What is this mRNA actually? Anyone? RNA.
Ma'am, it will synthesize proteins from
Uh-huh.
amino acids.
I'm
Yes.
not clear.
Ma'am, it will synthesize proteins from amino acids.
Yeah, yeah, that is okay, but structurally, structure-wise, I'm asking, what is an RNA? Structure-wise, I'm asking. See,
Ribonucleic acid.
yeah, see, DNA means you will be having a double strand. Okay, but RNA, it will have only one single strand. Understood? Okay, it will have only one single strand. In the case of DNA, you will have two strands intertwined with each other, right? But in the case of RNA, you will be having what? Only one single strand, where you will have a sugar phosphate backbone attached to 111 nitrogenous bases. Okay, but in the case of DNA, you had two. Okay, and those two actually, what will happen? That complement a nitrogenous basis, it will get attached. Okay, see in this figure, are you able to see? I have written it as, yeah, I will just see, are you able to see here? See here, you have a double helical structure, right? Right, see, it will be like this, right? You will be having here a nitrogenous base, here one nitrogenous base, here one, and here one. Okay, this is how your DNA looks like, right? In between, this will be bonded by what? Hydrogen bond. Okay, this is a DNA, but only this one side you take, only this one side. So it's just a single strand. Okay, it's just a single strand. This is a sugar phosphate backbone having one nitrogenous bases. Okay, this is what is called as a RNA. Understood what the difference between DNA and DNA and RNA? Just only one strand with nitrogenous bases is what is called as an RNA. See now, suppose if the body if you're getting if an information is given to the cell that you have to produce insulin immediately. Okay, because the body needs insulin now. Okay, what actually it will happen? We have studied, no, in cell membrane, protein signal proteins will be there, that actually it will receive the signal and it will give the information to the cell. Okay, that you have to produce insulin immediately. The information, it will reach the nucleus. Okay, so in the nucleus, whichever DNA, it is going to have the code to produce insulin. Okay, that particular DNA, it will start getting replicated. Okay, so what is mean by replication? Means this DNA which is going to have a double strand, no, that it will be broken down, okay, by means of RNA polymerase. A copy, okay, a copy of this DNA, it will get produced. That is what is called as what? Replication. Understood? So whichever DNA, whichever part, okay, whichever it's going to have that code for insulin, that it will undergo replication. That means a copy of this information, whatever it is stored in the DNA, it will be produced as what? mRNA. Understood? This is called as a pre-mRNA, but later it will have some structural changes, it will happen, and finally you will have what? mRNA. So what does this mRNA means? It's called as messenger RNA. See, I have told you there are three types: mRNA, rRNA, and you will be having one more thing called as tRNA. All the three are involved in the synthesis of protein. One is called as messenger
And what
RNA.
is rRNA, ma'am?
I will tell you. mRNA is the messenger RNA. Okay, this is the one which has the code for what? That enzyme. Understood? Next, rRNA is called as ribosomal RNA. See, this ribosomal RNA and protein only together, it will form ribosomes. Okay, because ribosomes are very, very important, because that is the one which is going to read the codes. See, I told you, no, here, 3 I will draw one diagram. This is an mRNA. This is a nitrogenous base. See, it's like this, right? It may be A, okay, it may be T, it may be G. See, I think in RNA, one of the nitrogenous bases, it's a different. Okay, I think it's something C. Okay, like this it is there. Okay, this 3 is what is called as a codon. Understood? 3 nitrogenous bases, it will be called as a codon. So this ribosome is the one which is going to read that particular codon, and an amino acid corresponding to that, it will be picked. See, in the cytoplasm, you will be having so many amino acids. Okay, it will be picked by what? tRNA, which is called as what? Transfer RNA. Understood? All the three are involved in protein synthesis. Messenger RNA is the one which is going to have the information. Okay, codes. Okay, rRNA, it's ribosomal RNA, that is the site. Okay, ribosomal RNA, it's very, very important for the synthesis of what? Ribosomes. Okay, so what is the role this ribosome plays? This ribosome is the one which is going to read this code. Understood? When it reads this code, it will give the information, okay, to tRNA to pick that amino acid. So the tRNA from the cytoplasm, it will pick the amino acid. Okay, so here are you able to see, no, it will start from one point. Okay, they will call it as initiation point. So first, from the initiation point, first three codes, ribosome will read, tRNA will bring one amino acid. Okay, it's here. Then it will read another three. tRNA will bring another amino acid. Like that, it will keep on getting formed. Okay, finally, when the information is over, okay, because it's just a strand, right? It's a segment of a DNA, right? When all the amino acids gets collected, it undergoes some structural changes to finally to get what? Insulin. Understood? So this is how actually a protein is produced. Okay, now it is 23, right? See, now let me play that particular video also. You just go through that video. Okay, even today also, even when you are having your leisure time, go through that video, and tomorrow's class we will discuss about it. Okay, just an idea I have given you. In between
Will you
video,
send the link for the video, ma'am?
yeah, I will do that.
Okay.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription.
See, are you able to understand how actually this mRNA is getting produced? See, you had two strands of DNA, okay? Then an RNA polymerase, when it gets attached to the DNA, it just gets split, okay? And one strand is exactly copied. Copied means what is happening? See, suppose if A is there in the DNA polymer, what is a complementary to A? It is T, right? So one of the T nitrogenous bases, it will just get attached. Next, if it is C, then G will get attached. Okay, like that, a complementary to the nitrogenous bases, whatever was there in one strand, okay, exactly opposite to that it is formed, and that finally it gets separated out as what? mRNA. Understood? Now you have the messenger
Before the
RNA.
messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA.
See, actually, if you would have seen in the diagram, there is something called as pre-mRNA and mRNA, because not the entire stretch of the RNA will be useful. Whichever it is useful contains the actual code, that alone it will be taken, and the remaining it will just get what? Removed. Okay. So the final RNA, whatever it will be there, it is called as what? mRNA. Okay, the other one is called as, the one exactly copied from DNA is what is called as pre-mRNA. Okay, then the removed unwanted segments after removal, it will be called as what? RNA, mRNA.
Messenger RNA then moves out of the nucleus into the cytoplasm.
Okay, now through pore it is coming to the cytoplasm.
Protein factories in the cytoplasm, called ribosomes.
Okay, now these are the two ribosomes. Okay, one a larger unit and one a smaller unit of ribosome. It will get attached, okay, to that RNA, and this is the one which is going to read three nitrogenous bases at a time, called as codons. Okay, so what is this ribosome made up of? It is made up of rRNA and protein. Okay.
Bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome.
Say, are you able to understand what is tRNA? See, this tRNA, it will now have three nitrogenous bases which will be complementary to whatever it is there in the mRNA. Okay, that only it will exactly fit. If the order is changed, means it will not fit. So this UCA means there it will be having it as what? CUT, like that. Okay, so that it will have a specific amino acid. So that amino acid, it goes and gets linked to that. Okay, like that only the entire segment it will be read. That is the code, it will be read three at a time, and the specific amino acid it will keep on getting joined to finally produce what? A polypeptide chain.
The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Understood? This sequence, it will undergo all the structural changes, whatever it is needed, and finally it will become a protein. Is it clear?
Ma'am, you will share this video's link.
Yes, ma'am.
Video link I can give, video as such I can't. Okay,
Yes.
video link I will give you.
Yes, ma'am.
Yeah.
Will you post it in the WhatsApp group or you will post it here only, ma'am, in Teams?
In Teams only, in post I will give. Okay.
Okay.
I will try to do that in post. Okay, the link I will try to post, because here in that file folder I will not be able to upload videos. Okay, so videos link, if you want, I will just give it. These links I will give you, because anytime it will be very helpful. Easily you can understand, because you can have a view, right? So easily you can understand. So now is it clear? Anything, any doubt you have regarding this gene, chromosome, chromatin, proteins, all those things?
Ma'am, can we read through the PPT once and rewatch these videos and get back with the doubts in the next class?
Yes. That's what even I want, because now it's yeah. Yeah, no problem. Okay, you do that, huh? Uh, because it's not that difficult also. If you see the video, if you see the PPT, easily you will be able to understand. Okay, see now what I want you to tell is you have DNA, okay, double strand. But what I want you to do at the same time is don't go too much into this. Then it will get highly complicated. Understood? So simple thing, you have a DNA. This DNA, when suppose a protein, if it has to be synthesized, a copy of this is made. Okay, you know how the copy it is made. How it is made means at that time, actually, when in an RNA polymerase, when it gets attached, the strands it will split into two, and exactly a complementary basis. Okay, complementary to whatever it is there in the DNA, it will form what? Pre-mRNA. This pre-mRNA, it will be whatever, only the functional part it will be taken, and finally that it is going to constitute your mRNA. This mRNA, see, till mRNA, where actually the process is happening, it is happening inside the nucleus. Okay, so this mRNA then comes outside the nucleus through the port to what? Cytoplasm. So protein synthesis, exactly what is it's happening in what? Cytoplasm. Okay, so in cytoplasm you will be having ribosomes. So this mRNA will get attached to the ribosome, and these ribosomes it will read three bases at a time. Okay, that is called as a codon. As it reads a specific tRNA, exactly complementary to the code. Okay, they will be called as anticodons. Okay, that it will carry an amino acid and that it will come and get linked to this mRNA. Like that, as the code is, as the ribosomes read the code, 11 tRNA, it will bring the, what is that? Exact amino acid corresponding to that code, and finally all these amino acids, it will form a polypeptide chain. Okay, then this it will undergo a structural changes to give you what is called as the protein. Okay, so this is one important function of the nucleus. Okay, next it controls the structure of the cell by that is what, transcribing DNA, which encodes for structural proteins like actin. See, almost all protein, okay, exactly, whether it is actin or keratin or other insulin, whatever may be the protein, the process is going to be the same, but the DNA will be different. Okay, so the nucleus is the site of ribosomal RNA. Okay, see, this ribosomal RNA is important for the construction of what? Ribosomes. Okay, ribosomes are made from ribosomal RNA, and this ribosomal RNA is synthesized where? It's inside the nucleus, and particularly inside nucleolus. Okay, normally they will call nucleolus as what? The center of ribosomes. Okay, because nucleolus is the place where this rRNA will get synthesized. Okay, so this rRNA together with some protein only is your ribosome. Okay, and why ribosome is important? You know, ribosome is the one which is the site of protein synthesis. Okay, this is the one which is going to read the codon and bring and make the exact tRNA to come bring the specific amino acid. Okay, then characteristics are transmitted from parent to offspring through genetic material contained in the nucleus. Okay, so just in simple words, if I have to tell, means it controls the genetic information of the cell and the heredity of an organism. Okay, so it controls protein enzyme synthesis, it controls cell division, cell growth. Okay, it stores DNA, RNA, ribosomes, etc. Okay, and it regulates the transcription. See, next class I will tell you which is transcription, which is translation and all. Okay, regulation of the transcription of mRNA into protein and production of ribosomes. Okay, so these are all the important function of nucleus, and that is why this is called as what? The brain of the cell, because it controls each and everything, right? So nucleus, so this is going to be very, very important. Okay, so better now let me stop with this. Next, tomorrow's class, I will once again, because I will start with ribosomes. Okay, again in ribosome also the same thing is what we are going to see. Okay, so for that particular reason only, I'm just telling you, go through. See, here also, I will tell you about all these things only. Okay, so for that purpose, go through the video. Now itself, I will try to give the link. Go through the video. Okay, go through the PPT and just come. Okay, if you are having any doubt regarding this, get it clarified. Okay, and even if you have any doubts now also, you can. Because it's all a little bit new to you people know. Once if you go through and if you see the video once again, means you will be able to understand. Okay, yeah, so if you have any doubts.
Ma'am,
Hmm.
in RNA we have uracil, no, instead of thymine.
Yeah, that is what I told you. See, these are the two main differences. Okay, the one single strand, but in the case of RNA alone, see, normally in DNA it is adenine, guanine, thymine and cytosine. Okay, but here in this case, in the case of RNA, instead of thymine they will call it as what? Uracil.
Okay, thank you.
Yeah. But four nitrogenous bases only, but in RNA only one of the thing it will be replaced by uracil. But it plays the same role, same thing. See, listen, tomorrow's class, definitely I'm going to ask you question and then only I will proceed. Okay, any one I will just ask. but just because of this reason, just don't absent yourself for the class. Okay, see, this is the way how you will learn. Okay, just simply say just attend a class, see some videos, and then if you keep on going means it's waste. Okay, you have to get some idea out of this. Okay, so tomorrow's class particularly, why? Because means it's important. Okay, you should know this. That is why I'm telling you, just go through and come. Okay, let me have it as a sort of a discussion class tomorrow. Okay.
Only for this class's topics or all that were taught before this?
Particularly this for tomorrow, I'm telling. Every class, if it is like that, means it's good only, no?
Yes, ma'am.
Yeah, particularly this, you just go through and come because it's all new. So many things are there in that. So that is why I want you to read once and then come. Okay, tomorrow I'm having first hour class. So just go through once and come.
Okay, ma'am.
So if you're not having any doubts, yeah, you can leave. Thank you.
Thank you, ma'am.
Yes, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you,
Thank you,
ma'am.
ma'am.
Thank you.